All right, everyone, welcome back. Today's session is a deep dive into a topic that bridges foundational microbiology with some of the most exciting therapeutic frontiers in medicine, phage biology and phage therapy. We've called this talk The Return of Nature's Bacterial Predators, and that's not just dramatic flair. We're genuinely watching a resurgence of interest in bacteriophages as a solution to one of the biggest challenges in medicine today, antibiotic resistance. We'll walk through how phages function biologically, their life cycles, their interactions with hosts, and how they've co-evolved with bacteria. And then we'll shift gears into phage therapy, what it is, why it's important, and how it's being retooled in the modern clinical landscape. So, why revisit phages now, after almost a century of ignoring them in mainstream Western medicine? The answer is right there: multi-drug resistant, MDR, and extensively drug-resistant, XDR, bacteria. These are pathogens that have acquired resistance to most, if not all, classes of antibiotics. They're not just theoretical threats anymore. They're already in ICUs, on surgical wards, and increasingly in community settings. Phages were actually discovered before antibiotics. In the early 20th century, they were seen as miracle cures, but with the discovery of penicillin and other antibiotics, interest in phages waned, especially in the West. In contrast, countries like Georgia and Russia kept researching and using them clinically. Now, with the antibiotic pipeline drying up and resistance skyrocketing, phages are getting a second look, not as replacements, but as potent allies in our antimicrobial arsenal. Let's step back and define our subject. Bacteriophages, often just called phages. These are viruses that specifically infect bacteria. They're ancient, they're everywhere, and they're the most abundant biological entities on Earth. We're talking orders of magnitude more than all other organisms combined, around 10 superscript 3 superscript 1 of them. Structurally, many phages have that iconic spaceship shape, a head or capsid containing nucleic acid, a tail used to inject DNA or RNA into the host, and sometimes tail fibers that help find and latch onto the right bacterial cell. But phages are diverse. Some are filamentous, some have lipid envelopes, and their genetic material can be DNA or RNA, single or double-stranded. Their abundance and specificity make them both fascinating ecologically and powerful therapeutically. Let's look at the lytic cycle. This is phage warfare at its most aggressive and clinically useful. A lytic phage lands on a bacterium, injects its genetic material, and completely hijacks the bacterial machinery. The cell stops doing its usual work and instead becomes a phage factory. New viral particles are assembled, and eventually, the bacterial cell bursts open, lyses, releasing new phages to go infect others. This is the kind of phage we want for therapy: direct, targeted bacterial killing. The cycle is fast and lethal, making lytic phages excellent candidates for treating active infections. Now let's contrast that with the lysogenic cycle, a more subtle, sneaky approach. In this cycle, after a phage injects its DNA, it doesn't immediately hijack the host. Instead, it integrates its DNA into the bacterial chromosome. The host cell keeps living and dividing, unknowingly copying and passing on the phage DNA, now called a prophage. The phage can remain dormant for many generations. But under stress, such as UV light, antibiotics, or DNA damage, the prophage can activate, excise itself from the genome, and enter the lytic cycle. It's like a sleeper agent inside the bacterium. From a therapeutic standpoint, this duality makes temperate phages, those capable of lysogeny, less ideal for direct use. But they're important for understanding bacterial evolution and gene transfer. Here's the clinical bottom line. When we're using phages to treat infections, we want phages that are strictly lytic.
Why?
Because lytic phages do their job. They infect and kill bacteria without sticking around or meddling with the bacterial genome. Lysogenic phages, by contrast, carry risk. Not only can they lie dormant, but they can also confer new traits to the host bacterium, including virulence factors and resistance genes. That's a big red flag in a therapeutic context. So, for safety and efficacy, lytic phages are the clear choice, and phage therapy pipelines invest heavily in screening out lysogenic behavior. This slide touches on a critical and often misunderstood aspect of phage biology, their potential to enhance bacterial virulence. Some phages carry genes that, when inserted into the bacterial genome, make the bacterium more dangerous. This is called lysogenic conversion. Classic examples include the cholera toxin in Vibrio cholerae and the diphtheria toxin in Corynebacterium diphtheriae. Both are phage-encoded. Phages can also carry antibiotic resistance genes, acting as mobile genetic elements. This is why temperate phages are scrutinized heavily before any therapeutic application. We don't want to accidentally turn a harmless commensal into a superbug. One of the defining features of phages is their extreme specificity. A single phage might infect only a specific strain of a species, which is both a blessing and a limitation. It means we can avoid damaging the rest of the microbiome, no collateral damage like we see with broad-spectrum antibiotics. But it also means that identifying the right phage for the right pathogen, what we call phage matching, is crucial. Bacteria aren't passive either. They evolve resistance to phages by mutating receptors, modifying their cell surface, or using CRISPR-Cas systems. In turn, phages evolve to overcome those defenses. It's a microbial arms race, and it's part of what makes this field so dynamic. So, what is phage therapy, practically speaking? It's the use of these viruses to treat bacterial infections. We can administer phages in several ways: topical for wounds, oral for gut infections, inhalation for respiratory pathogens, and intravenous for systemic infections. One of the coolest aspects is that phages multiply only in the presence of their host bacteria. That means they amplify themselves at the site of infection and taper off when the job is done. It's like an intelligent drug that adjusts its dosage in real time. This slide really gets to the heart of why phage therapy is worth our attention. First, phages are narrow-spectrum. They leave your beneficial microbes alone. That's huge, especially for patients with delicate microbiomes or who are prone to secondary infections after antibiotic treatment. Second, phages are good at penetrating biofilms, those slimy, protective layers bacteria form on surfaces like catheters, implants, and wounds. Antibiotics often can't reach inside those, but phages can. And finally, phages evolve. If resistance arises, we can isolate a new phage or engineer an existing one. It's a living, adaptive therapy. But we're not quite ready to roll phages out as standard therapy yet. Their narrow host range means we need precise diagnostics first. We have to know exactly which pathogen we're dealing with. Regulatory frameworks are still evolving. Is a phage a drug? A biologic? Something else? Also, our immune systems might neutralize phages before they can act. And large-scale production has to be safe, free of endotoxins, and consistent in quality. These are all active areas of research and development. Despite the hurdles, we're already seeing phages move from theory to therapy. The 2016 case of a patient with a drug-resistant Acinetobacter baumannii infection who was saved by an emergency phage treatment, that was a turning point. Since then, other case studies and small trials have followed. We're now seeing phages used under compassionate use protocols and even entering clinical trials. And this is just the beginning. Now, this is where it gets futuristic and exciting. Synthetic biology allows us to edit phage genomes, for example, to remove lysogenic genes or add features like enzymes that degrade biofilms. A CRISPR-equipped phages can go even further, deleting resistance genes from bacteria, essentially making them vulnerable again. This level of control transforms phages from natural predators into programmable antibacterial tools. What does the future look like? We'll likely see more phage-antibiotic combinations tailored to each infection. Agricultural use is also rising, using phages to control foodborne pathogens in poultry, fish, and vegetables. Phage banks, searchable libraries of validated phages, are already being built. These could make personalized phage therapy a reality in the near future, particularly for chronic or resistant infections. To bring it all home, phages have been hunting bacteria for billions of years. We just happen to be late to the party. Their specificity, adaptability, and potency make them perfect complements to our existing antibiotic strategies. As we face an age where antibiotics are no longer guaranteed to work, phages offer a ray of hope, a biological solution to a biological problem. Thanks for staying with me through that whirlwind tour. I hope this gave you not only a refresher on phage biology, but also a window into where the field is going. Let's open the floor for questions, thoughts, or even debates. I'd love to hear your take on the clinical, ecological, or biotechnological aspects of this topic.